GR team presenting in breast cancer webinar. Today, I am presenting the activating CEBPα with a small activating RNA drug for triple negative breast cancer. Breast cancer is most common cancer in women. Triple negative breast cancer, called the TNBC, is characterized by the loss of the estrogen receptor, progesterone receptor, and the human epithelial growth factor receptor 2, accounting for the about 15 and the 20% of the total number of the breast cancer diagnoses. However, TNBC shows a high response rate to systemic therapy compared with other breast cancer subtype. The tumor differentiation of the TNBC is poor, with an early tendency to metastasis and a higher recurrence rate. Relapses and the metastatic TNBC usually progress more rapidly, showing strongly resistant to chemotherapy and the radiotherapy. Due to the lack of the combination target drug, alternative treatment failed to improve the disease patient's prognosis and the quality of life. Finding the drug of the TNBC is the critical of patients with advanced TNBC. Today, I am presented the potential gene therapy drug for the TNBCs. Next slide shows an overview of the research and the development of TNBC targeting drug registered in the clinical trials. There are approximately 707 clinical trials for TNBC treatments registered in the database of the clinical trial. Pie chart summarizes the current research and the development progress of the targeted drug for the TNBCs. Drug-targeted classic cell signal cascade still dominated the majority of the TNBC drug. Yellow pie chart of the immunotherapeutic drug. Despite the significant drug in the TNBC treatment, the TNBC remained without main effectively therapeutic as it is a very aggressive cancer. The CEBPα is a major regulator of the lymphoid lineage and the myeloid differentiations. CEBPα has published the anti-cancer drug as a tumor suppressor in various tumors and is involved in the regulation cancer cell differentiation, proliferation, apoptosis, and the metabolisms. Unregulated of the CEBPα has the potential to improve the liver function and limited the hepatocellular carcinoma growth. Small activating RNA drug called the siRNA, specially targeting the CEBPα, formulated in the enzymatic liposome nanoparticle. CEBPα targeting the siRNA can restore the endogenous CEBPα expression and shown to lead to the better therapeutic outcome in cancer. Next, we confirm the CEBPα expression in cancer depend on the mRNA expression database. It is generated by CEBPa. As you can see the graph, CEBPα expression is inhibited in breast cancer. The breast cancer has a different subtype. They called basal-like, HER2, luminal A, and the luminal B. Basal-like type breast cancer is similar with the TNBCs. We analyzed the CEBPα expression in subtype of the breast cancer. The level of CEBPα mRNA markedly reduced in the TNBCs. This result suggests that reduced the expression of the CEBPα may play a role in the development and the progression of the breast cancer. This heat maps shows the gene expression is significantly associated with subtype. This result generated by UCSD-GENA browser. This browser can allow the explanation the functional genomic data set. Red is the represented the high expression, and the green and the blue means the low expression gene in the breast cancer. Each row shows the breast cancer cell lines, cells were classified the basal. They just presented their BA or BAB, and luminal LU represented the purples. The basal subtype constitute 20% of the breast cancer, such as keratin 5 and 17, amine, and the fatty acid binding protein 7, and low expressed of the luminal genes. The 80% of the basal-like cancer are triple negative in nature. Basal-like type shows the low expression of the CEBPα. This picture drew in previous slide. The major TMPC cell line of the SUM159 and the MDA-MB231 belong to basal-like type. CEBPα expression is down-regulated in the TMPC cell lines. We are curious about gene expression in the breast cancer patient. Red arrow showed the metastatic tissue. Zoom in the heat map, normal solid tumor showed the high expressed the CEBPα genes. However, we find the CEBPα down-regulated in metastatic tissue from the breast cancer patient. Extracellular signal of the TGF-beta can induce the epithelial to mesenchymal transition in cancer of epithelial origin, and promoting moleculars and the phenotyping change resulting in pro-metastatic characteristics. This publication shows the mesenchymal marker of the N-cardiorin, fibronectin, or preventing increasing in the TGF-beta treatment. The epithelial marker of the E-cardiorin is reduced. As you can see the cell picture, TGF-beta treatment to show the cell shape of mesenchymal pictures. Interestingly, CEBPα mRNA expression was rapidly reduced upon the TGF-beta treatment. Next, to confirm CEBPα leads to epithelial phenotype, they did the experiment of epithelial cell culture system as a three-dimensional spheroid. Left slide picture showed the TGF-beta stimulated reduced the spheroid numbers. CEBPα gene activation shows the feature of the epithelial spheroid. Both treatment of the TGF-beta and the CEBPα induced is sufficient to drive the recognition of the spheroid architectures. This result suggests that activation of the CEBPα expression in mesenchymal population is sufficiently to revert this cell toward an epithelial state. CEBPα activation did not reduce in the tumor volume. However, long immunohistochemistry shows that reduced the number of the metastatic lesion in CEBPα overexpression group mouse. This result supports that the CEBPα may role for the anti-metastatic drug. Next, we evaluated the anti-inflammation potential of the CEBPα saRNA. As you can see this graph, the secretion of the pro-inflammation cytokine TGF-alpha and IL-6 and IL-1 beta significantly inhibited by the CEBPα saRNA. We confirmed that anti-inflammation activity of the CEBPα siRNA in vivo. We generated the LPS-induced the inflammation animal model in humanized mouse. The humanized mouse can harbor human cells and mimic the human immune systems. This system can provide a better understanding of the human immune response and the effect of the biological therapy during the LPS-induced the inflammation. LPS is the liposaccharide. LPS is a major outer membrane component of the gram-negative bacteria. It is able to induce an inflammation response in activation human immune cells. LPS stimulation releases the multiple pro-inflammation cytokines in vivo. After generation, LPS humanized mouse model, we tested the gene expression level from the mouse blood sample. LPS stimulation suppressed the CEBP-alpha mRNA level. Injecting the MPL CEBP-alpha restored the CEBP-alpha expressions. The activation of the CEBP-alpha gene led to the decline of the immune and the inflammatory response gene of the CEBP-beta, CXC motive chemokine receptor 4, and the programmed DAS1 ligand. Next, we determined the inflammatory response of the MPL-CEBP-alpha activation in the LPS humanized mouse from peripheral blood and plasma. MPL-CEBP-alpha treatment resulted in decline of the pro-inflammatory cytokine. This result suggests MPL-CEBP-alpha effectively repressed the LPS-triggered inflammation response in vivo. Next, we determined the CEBP-alpha can work for the potential drug of the TNBC. Firstly, we detected the gene expression and the cytokine level in the breast cancer. Non-TNBC cell line of the MCF7 is highly expressed the CEBP-alpha, but TNBC cell lines showed the low CEBP-alpha expression level. The expression of the pro-inflammation cytokine of the IL-6 and the IL-8 and mesenchymal marker of the protein pimentin regulated in the mesenchymal-like TNBC cells of the MDA and B231s and the SW159s. Next, we confirm the CEBP-alpha siRNA efficacy in the breast cancer cell lines. siRNA activated the CEBP-alpha expression in the TNBC cells, but non-TNBC cell of the MCF7 did not change the CEBP-alpha expression level treated with the siRNAs. This result shows that CEBP-alpha siRNA can specifically activation the CEBP-alpha gene in TNBCs. We confirm the pro-inflammatory cytokine expression to detection IL-6 and the IL-8 expression level in breast cancer. The MCF is the non-TNBC cell line did not express the IL-6 and IL-8. The increase in the CEBP-alpha is associated with reduced the pro-inflammatory cytokine in MDA and B231. We can see previous slide. The analysis of the TCGA database in the TNBC cell lines and breast cancer patient showed the IL-6 and the IL-8 is a critical pro-inflammatory cytokine in TNBC cells and the metastatic tissue sections. The cytokine is unregulated. We determined the secretion of the IL-6 and the IL-8 in TNBC cells. IL-6 and the IL-8 decreased the secretion when we treated the siRNA. The basis of our result showed the IL-6 and the IL-8 are critical factor. CEBP-A siRNA can reduce the secretion of the IL-6 and the IL siRNA provide the TNBC anti-pro-inflammation activities. We transfected the siRNA into the MDA and B231, SW159, and the MCF7 is the non-TNBC cells, and then cell viability measured by the MTT assay. The proliferation of the MDA and B231 and the SW159 cells are decreased the following treated with the siRNA. However, the proliferation of the MCF7 is the increasing with the siRNA treatment. This observation suggests the CEBP-alpha siRNA specifically inhibited the cell proliferation in TNBC cells. In order to get a TNBC tumor model, we generated the TNBC xenograft model in NSG mouse. TNBC tumors rapidly grow 4 weeks later. To identify MPL-CEBP-alpha can inhibit the tumor progression, we injected the MPL-CEBP-alpha intratumorally into TNBC xenograft model. MTL CEBPα reduces the TNBC tumor growth, but this inhibition is not significantly compared with the control groups. Next, we thinking about how we improve the anti-cancer and the anti-metastatic effect in the TNBCs. Pimentin is one of the most major factors associated with epithelial mesenchymal transition called the EMT. EMT is the loss of the epithelial characteristics and the acquisition of mesenchymal phenotypes. The mesenchymal phenotype increases the TNBC migration behavior. So, EMT can play a relevant role in tumor invasion and the metastasis. The elevation expression of the pimentin contributed to the aggressive phenotype and the poor prognosis in TNBCs. We generated the pimentin targeting siRNA and tried the combination therapy with CEBPα siRNA and the pimentin siRNA. The pimentin siRNA efficiently reduces the endogenous pimentin expression in MDA-MB231 and SW159. The IL-6 expression significantly inhibited the combination therapy compared with the treatment single siRNA. Next, we determined whether our therapeutic drug inhibited the ILA production in TNBCs. We treated the drug and measured the ILA secretion. The combination therapy of the siRNA and the siRNA can reduce the ILA secretion in TNBC cell lines. This picture shows the tumor microenvironment in the breast cancer. Tumor microenvironment in the breast cancer contributed to the stem cell-like properties and the immune escape of the tumor. TNBC is highly infiltrated by the cancer-associated fibroblast, and the lymphocyte can secrete the CXCL12 to promote the resistant phenotype of the TNBC cells and to recruit the T-regulated cells to suppress the immune response of the infected T cells. In our experiment, increasing the CEBPα by the siRNA can deactivate the anti-inflammatory response, and the epithelial mesenchymal transition promoted the TNBC metastasis. We think the combination therapy of the CEBPα siRNA and the pimentin siRNA supports the possibility anti-metastasis drug. Thank you for your attention, and I especially thank you for my mentor of Dr. Rossi, and I appreciate the Dr. Rossi lab member and the collaborator, Dr. Habib, for powerful discussion with me. If you have any question, feel free to give me. Thank you.